Welcome back to the Deep Dive, where we sift through the most complex, cutting-edge research to give you, the strategic and technical foundations you need to understand tomorrow's medicine. Today, we are undertaking it's extraordinarily ambitious deep dive. We're looking at a concept that really pushes the boundaries of fetal intervention in utero heart transplantation, or IUHT.
And we should be clear from the start, what we're examining today isn't some speculative thought experiment. It's a structured review from Dr. Boris Petrakovsky and Dr. Lev Nemotin.
Right.
And they frame IUHT as, and this is their term, near-future medicine. It's all grounded in a really robust, conceptual, ethical, physiologic, and translational framework.
So their mission is basically to show that this isn't science fiction anymore.
That's it, exactly. They want to demonstrate that if you look at the convergence of existing advances in fetal surgery, in immunology, in donor ethics, all the pieces are there to make this scientifically feasible.
And the clinical need driving this, it's just tragically clear, isn't it?
It's profound.
We're focusing on lethal congenital heart defects. These are conditions that, they doom the fetus to death shortly after birth.
Right. And the authors, Petrakovsky and Nemotin, they cite some pretty stark data. We're talking about conditions like severe hypoplastic left heart syndrome, or HLHS, or maybe critical aortic stenosis.
Things that are just incompatible with postnatal survival.
Precisely. And they affect roughly 1 in 4,000 births here in the United States.
So you do the math on that?
It translates to nearly 1,000 fetuses every single year. 1,000 fetuses facing what is currently, for all intents and purposes, a death sentence.
Because the current option, neonatal heart transplantation, it just doesn't work for this population, does it?
It's completely inadequate. First, there's the scarcity of donors. Finding a heart from an infant that's the right size is incredibly rare.
And even if you find one, the baby's just so fragile.
Exactly. You have extreme hemodynamic instability, the overall fragility of a newborn with catastrophic heart failure. Most just can't survive long enough to get the transplant, or they can't tolerate the rigors of the procedure itself.
So IUHT is proposed specifically to address this huge unmet need.
Yes, by shifting the intervention into what they argue is a far, far more advantageous physiological environment.
Okay, let's unpack that. Let's get into this core scientific premise.
Yeah.
Because Petrakovsky and Nemotin are proposing a genuinely transformative shift in timing.
They are?
So what are these two massive, non-replicable biological advantages that IUHT leverages? What makes it, at least in theory, so much better than a neonatal transplant?
The entire strength of their argument, really, it rests on these two biological windows that you only get during gestation.
Right.
The first is what they call the natural cardiopulmonary bypass system.
The placenta.
The placenta. It provides continuous oxygenation. It removes all the metabolic waste. This stability completely, and I mean completely, eliminates the need for mechanical life support.
So no CPB, no ECMO.
None. You don't need a heart-lung machine during the actual heart replacement.
And that is a critical differentiation. Avoiding the heart-lung machine in a critically ill neonate means you avoid this whole cascade of catastrophic complications. We'll get into those later, I'm sure.
We will. It's a massive point. And the second advantage, which is really the immunological cornerstone of the whole thing, is fetal immune tolerance.
Right.
The mid-gestation fetus has this uniquely tolerogenic immune system. So if you introduce the donor organ during this very narrow developmental period, you can actually program the recipient's immune system to recognize that graft as self.
So no rejection, or at least a much lower risk.
It radically reduces the risk of long-term graft rejection, and it raises this incredible possibility of maybe, just maybe, eliminating the need for aggressive, lifelong immunosuppression drugs after birth.
We have to be clear here, though. We have to acknowledge the current state of play.
Of course.
The paper cites other reviews from Gardner and Yilmaz-Furton that confirm, as of right now, late 2025, no human IUHT has ever been performed.
That's correct. It's all been preclinical.
Current prenatal cardiac therapy is really just confined to structural fixes, right? Things like balloon valvuloplasty or putting in shunts.
Structural repairs, not whole organ replacement.
So what this paper is doing is charting a new course. It's saying, look, we know this hasn't been done, But here is the plausible pathway forward.
That's what makes it so compelling. It succeeds in integrating these established clinical facts, things we validated in other areas, like in utero hematopoietic stem cell transplantation, or IUHT.
Right. And complex fetal structural repairs.
Exactly. It connects those existing dots from across different disciplines to move IUHT from just a concept into a really actionable, phased translational roadmap.
Okay, let's deepen this discussion on that immune advantage. Because if this works, if you can actually achieve tolerance, it just fundamentally changes the entire prognosis for pediatric transplant recipients.
It's the centerpiece of the whole framework.
So why exactly is the mid-gestation fetus so immunologically perfect for transplantation?
It all comes down to exploiting this unique window of immunological development. The paper references principles that have been validated through years of IUHCT studies, including some of the pioneering work by Marianos, Heaton, and Flake.
And it's all about timing.
It's all about timing. The fetal thymus is actively developing its central immune competency. It's learning what's self and what's other. So if you expose it to foreign, non-pathogenic antigens during this phase.
You're talking about the donor cells.
The donor cells, yes. You program the immune system to just accept them.
So the fetus essentially learns to accept the donor tissue before its immune system is mature enough to learn how to reject it.
You've nailed it. And the outcome is what we call the induction of stable microchimerism.
Can you break that down for us, microchimerism?
Sure. It just means the long-term, low-level persistence of donor hematopoietic stem cells, the cells that make blood and immune cells within the recipient.
So a little bit of the donor lives on inside the recipient forever.
Exactly. And these donor cells are continuously presenting their unique antigens, which creates a state of donor-specific tolerance. It's like you're tricking the developing immune system into treating the donor organ and all the donor cells as just a normal part of the body.
And if you can achieve that state of tolerance successfully, what does that mean in practical clinical terms down the road?
I mean, it means you could potentially bypass all the severe chronic complications that come with lifelong immunosuppression. The absolute worst long-term outcome in pediatric heart transplantation is something called cardiac allograft vasculopathy, or CAV.
Okay, define CAV for us. That sounds like a critical term here.
It is. CAV is often called the Achilles heel of long-term heart transplant success. It's a really aggressive, accelerated form of coronary artery disease.
So the arteries of the new heart get diseased.
Right. The coronary vessels of the transplanted heart thicken and narrow because of this chronic low-grade immune attack. It's a slow-motion rejection.
And what's the outcome of that?
It often leads to graft failure within just 5 to 10 years. It's a key driver of the incredibly high retransplantation rate you see in pediatric patients.
But by establishing tolerance in utero.
The IUHT framework aims to sidestep the very mechanism that causes CAV in the first place. You avoid the problem entirely.
The contrast with postnatal treatment is just so stark when you put it like that.
It's night and day.
Postnatal transplant kids, they rely on these really toxic, aggressive drug regimens. Calcineurin inhibitors, antimetabolites.
And the side effects are brutal. They cause renal toxicity, growth delays, neurocognitive issues, and hugely increased risks of aggressive lymphomas and infections.
So the ability to avoid that entire lifetime of biological burden by getting tolerance in utero, it shifts the procedure from being merely life-saving to being genuinely life-enhancing.
It fundamentally changes the risk-benefit equation. The goal isn't just a heart that beats. The goal is a biologically integrated organ that minimizes long-term metabolic disruption and actually enhances developmental normalcy.
It's about quality of life, not just survival.
Exactly. The long-term stability you get from tolerance offers a magnitude of benefit that, a simple mechanical or structural repair can't even touch.
Let's move to that second major advantage then.
Yeah.
The placenta's role. We mentioned avoiding mechanical life support. What exactly is the mechanical problem with operating on a critically ill newborn who's in profound heart failure?
The mechanical challenge is just overwhelming instability. To do the operation, you have to stop the failing native heart, which means you have to maintain circulation artificially using CPB or ECMO.
The heart-lung machine.
Right. And these mechanical systems, they introduce a huge amount of foreign surface area into the bloodstream. That triggers a severe systemic inflammatory response.
The body basically attacks the machine.
It does. And on top of that, these machines induce profound shear stress on the blood cells, causing hemolysis. The cells just break apart. That increases the risk of both bleeding and clotting, which very frequently leads to a stroke or early graft failure.
So the whole environment is just high risk, unstable, and metabolically incredibly stressful for a tiny baby.
It's a controlled catastrophe, really.
And this is where IUHT uses the placenta as this kind of elegant biological solution. How does it work? How does it maintain circulation during the procedure?
The placenta is just doing its job. It's performing continuous gas exchange. It's removing waste. It essentially ensures the recipient fetus stays perfectly perfused and oxygenated throughout the entire operative window.
So the surgeons are working in what the authors call a calm, consistent circulation.
That's the perfect phrase for it. The placenta stabilizes heart filling. It minimizes fluctuations in blood pressure, and it dramatically reduces the metabolic stress on the rest of the fetal body.
But does the heart replacement surgery require no perfusion assistance at all? It seems unlikely.
No, not entirely without assistance, you're right. The source material is clear that IUHT may still need a low-flow fetal perfusion circuit.
Okay.
This is a conceptual device we'll discuss later, the PSMU. And this circuit would be necessary for localized support right during the vascular cross-clamping phase, when you're actually implanting the donor organ.
But this is critically different from full neonatal bypass.
Orders of magnitude different. The scale is minuscule, the flow requirements are minute, and because the placenta is handling the main job of systemic oxygenation, the risks from this little circuit are just so much lower than what you see with full mechanical CPB.
And we know the fetus can tolerate this kind of thing, right? The stability offered by the placenta isn't just a theory.
Not at all. We have proof of concept from other complex fetal procedures, things like spina bifida repair or ex-site procedures, that's ex utero intrapartum treatment. They already demonstrate that the fetus can tolerate controlled, prolonged interventions while staying stable on placental support. The physiological underpinning for this is not hypothetical. It's grounded in decades of real-world fetal surgical experience.
Before we jump into the modern technology that makes this plausible, we have to recognize the sheer audacity of this idea by tracing its lineage. Let's talk about the foundational contributions of the Russian surgeon Vladimir Demikhov.
Demikhov's work is absolutely the essential historical context here. He was operating in the Soviet Union between the 1940s and the 1960s, and he achieved what seemed completely impossible at the time.
These were experiments on dogs mostly.
Yes, often on dogs. And they involved the first successful intrathoracic organ transplants in mammals. He wasn't thinking about immunology or fetal tolerance at all. His work was purely about anatomical and mechanical feasibility.
And what did he demonstrate that was so revolutionary?
He proved decades before Christian Barnard did the first human transplant that you could surgically remove a heart from one organism, transplant it into another, connect it to the circulation, and it would sustain life.
He had some pretty shocking experiments, didn't he?
His most famous, and to be honest, shocking experiments involved transplanting a second heart into the chest of a dog, creating a dual heart circulation. He even transplanted a head and forelegs, showing systemic integration.
Wow.
His work established the original proof of concept that cardiac replacement was, at a bare minimum, anatomically viable.
That's a staggering historical footnote. It proves that the core surgical challenge, just reconnecting the big vessels in a foreign body, was conquerable.
Absolutely. Demikhov solved the fundamental question. Can a transplanted heart be functionally integrated? So modern IUHT really stands on his shoulders. The frontier today isn't anatomy anymore.
It's immunology and miniaturization.
Exactly. It's about overcoming those remaining challenges, turning his initial audacious vision into a safe, reliable, and immune-tolerant procedure.
Okay, let's move forward. The physiological benefits are clear. But for IUHT to become a clinical reality, all the technical barriers have to be addressed.
Systematically adjust, yes.
The authors, and they reference pioneers like Evans, Harrison, and Adzik, they maintain that the evolution of fetal surgery over the last 40 years has made this core technical challenge approachable.
The transformation has just been revolutionary. The biggest initial barrier was simply getting to the fetus safely.
You mean access?
Access. Early fetal surgery required massive open hysterotomies, huge incisions into the uterus, and these carried significant maternal morbidity. We're talking massive blood loss, high infection rates, and a high risk of uterine rupture in any future pregnancies.
So what's the current state of fetal surgery? How has that changed?
We've decisively transitioned to minimally invasive fetoscopy and these hybrid open fetoscopic approaches.
So we're talking about tiny ports and cameras.
Exactly. Techniques now use specialized ports, usually only 2 to 5 millimeters in diameter, all guided by ultrasound and high-resolution cameras. This shrinks the uterine incision to just a fraction of its former size. Belfort's work, for example, really highlights how this minimizes trauma, it reduces hemorrhage risk, and significantly lowers the likelihood of preterm labor.
So the maternal risk of access is now more comparable to, say, a standard C-section scar.
A low transverse C-section scar, yes. And that's what makes the procedure ethically justifiable from the mother's perspective. Her risk is minimized.
And we know this works.
We do. The success of procedures like fetoscopic amniotic band resection or laser coagulation for twin-to-twin transfusion syndrome, it proves that you can do precision surgery inside the amniotic cavity. That provides the foundation for controlled cardiac exposure.
Beyond access, visualization is just paramount, isn't it? Especially when you're operating on a tiny moving target. What are the modern techniques that help with planning and execution?
The surgeon's eyes have just gotten dramatically better. We rely heavily now on high-resolution fetal echocardiography. That gives you real-time functional assessment of the heart chambers and vessels.
So you can see what's happening live.
And Doppler flow analysis gives immediate feedback on blood velocity and filling dynamics. Crucially, we can now do 3D reconstructions and even 3D printing of the fetal heart and the surrounding vessels.
And that lets you plan everything out beforehand.
It allows for incredibly precise surgical planning and accurate sizing of the donor heart, which minimizes the risk of a mismatch before the operation even begins.
Okay, this is where it gets really interesting and technical for me. The challenge of miniaturization and this idea of a rapid surgical workflow. The placenta takes care of the big picture, the systemic support. But the actual moment of transplantation connecting the vessels, that is an unforgiving race against the clock.
It's the ultimate bottleneck. IUHT requires what we call a warm ischemia window of ideally less than 10 minutes.
Define warm ischemia for us.
It's the time the donor heart is without any oxygenated blood supply at body temperature. Beyond that 10-minute mark, the risk of irreversible myocardial cellular death just skyrockets.
And that damage leads to what's called reperfusion injury.
Right, which often leads to immediate primary graft failure. The heart just doesn't start up again properly. That 10-minute window is the core technical challenge that everything else has to be engineered around.
So historically, what was it that prevented surgeons from hitting that 10-minute goal in these tiny hearts?
Simply put, scale. Standard cardiac surgical equipment, CPB circuits, instruments, they were all just too big and too slow.
But that's changed.
It has. Advances in neonatal cardiac surgery have led to some truly miniaturized components. We now have micro-oxygenators designed specifically for fetal flows. We're talking less than 1 liter per minute. And critically small volume priming loops.
The authors mention that these components, while they exist conceptually, need this rigorous validation in something they call the placenta synchronized microperfusion unit, or PSMU.
Right.
Let's talk more about that PSMU concept, because if we're talking about a late gestation fetus, the patient might weigh, 1,500 grams? If you prime a normal bypass circuit with 100 milliliters of fluid, you're immediately risking fatal hemodilution.
You've hit on the central engineering difficulty right there. The PSMU has to operate within an extremely narrow margin. To prevent that catastrophic hemodilution and the electrolyte shock that would follow, the total priming volume of the circuit.
That's the amount of fluid needed to fill the pump, the tubing, the oxygenator.
Everything. That total volume has to be strictly limited to 40 milliliters or less.
40 milliliters. That is an immense engineering challenge. You're talking about highly specialized micro-scale components where the fluid dynamics are governed as much by surface tension as they are by flow pressure.
It's a huge challenge. And the PSMU also needs sophisticated control systems to maintain the flow range, which the authors estimate to be between 20 and 100 milliliters per kilogram per minute for targeted circulation.
And it has to mimic fetal conditions.
Precisely. It has to regulate oxygen saturation to fetal levels, which are naturally lower than postnatal levels. If you hyperoxygenate the fetal circulation during bypass, you risk vasoconstriction and lung injury. It's a counterintuitive but absolutely critical design constraint.
So the PSMU isn't just about keeping the fetus alive. It's about precisely mimicking the intrauterine environment to protect both the graft and the patient. Okay, so back to that 10-minute bottleneck. How do surgeons actually speed up the anastomosis process itself?
It demands what we call super microsurgical techniques. We're talking about tools and materials you can barely see with the naked eye. 11 neuro and 12 0 sutures, which are finer than a single strand of human hair.
And the source material also references some newer concepts.
It does. Things like end-to-end anastomosis couplers, which are designed to mechanically join vessels almost instantly. And then there are future concepts like laser-assisted vascular welding and bioadhesive technologies.
So the key isn't brute force, it's precision speed, probably through robotic assistance and advanced material science.
Yes. The conceptual operative framework they detail in the paper requires robotically stabilized microinstruments to secure the anastomoses. They even talk about using a fetoscopic oscillating microsaw to safely open the fetal sternum.
So it's this combination of miniaturized tools, advanced visualization, and rapid attachment technology that makes them think the 10-minute window is actually achievable.
That's the idea.
Okay, so given that we're talking about prolonged, complex surgery inside the uterus, they estimate 2 to 4 hours. Let's pivot to the absolute foundational ethical determinant here, maternal safety.
Right.
The authors state explicitly that IUHT has to demonstrate risk levels that are comparable to established open fetal surgeries.
Yeah.
What is the current assessment of maternal risk?
Maternal primacy is completely non-negotiable. That's established by guidelines from groups like NAFNet. You always have to consider the mother first because she's not the direct beneficiary of the procedure.
Okay.
So if you review the AHRQ technical briefs and analyses from people like Belfort, the general risks tied to modern fetal surgery include standard anesthesia complications and, historically, hemorrhage.
But you said earlier that minimally invasive access has really helped with that hemorrhage risk.
Significantly. Modern series show hemorrhage rates are down to less than 5% thanks to the small size of the fetoscopic ports. The primary ongoing risk is and remains preterm labor.
The manipulation of the uterus can trigger contractions.
Exactly. It's a major concern.
So how are centers managing that preterm labor risk, especially for a procedure as long as IUHT?
It requires a rigorous multimodal strategy. You have proactive administration of tocolytics, which are drugs designed to suppress uterine contractions. You have precise uterine cooling to reduce metabolic stimulation. And critically, you have to minimize the operative time, which just reinforces the need for that rapid workflow we were talking about.
The sources also mention some rare but specific risks for IUHT. Things like VTE or venous thromboembolism from being immobile for so long, and even, in exceptionally rare cases, the need for maternal CPB.
That risk of maternal CPB, though it is rare, is a very serious consideration. If there were a profound fetal bradycardia or uterine hemorrhage that required immediate sustained intervention, the mother might need transient bypass support herself.
And that carries its own risks.
It does. Hypotensive risks to both the mother and the fetus. However, when you compare the overall risk profile against the established safety record of other open fetal procedures, like repairs for congenital diaphragmatic hernia, the authors conclude that the cumulative risks, while significant, fall within accepted ethical boundaries.
Because the procedure is targeting an otherwise lethal condition.
Precisely. The alternative is certain death.
We have to turn now to the ethical history, because the road to infant transplantation is, well, it's scarred by controversy. The authors spent a lot of time on this, learning from the past, specifically that tumultuous era of the 1980s and 1990s, and they highlight the high-profile cases of baby Faye and baby Teresa.
These historical cases, and they were documented thoroughly by authors like Donovan, Leggins, Faust, and Powers, they revealed that the primary obstacles to infant transplantation back then weren't really medical. They were systemic and ethical.
So baby Faye in 1984, that was the xenotransplant case.
Right. She received a baboon heart. And the procedure was medically aggressive, it was ethically ill-defined, and it led to this widespread public alarm and intense scrutiny of consent processes.
And then the case of baby Teresa, the encephalic infant donor, in the early 90s.
That case created an immense ethical firestorm, and it was all about the determination of death. Anencephaly is a condition where the fetus is born without parts of the brain and skull. Postnatal life is impossible.
But the baby has a beating heart.
Exactly. The parents wanted to donate her organs, but Florida law at the time required circulatory death. Because the organs were only viable if harvested while circulation was maintained, you had this absolute conflict. The child had a beating heart but no cognitive function. The law prevented organ donation, prioritizing a legal definition of death over the possibility of saving other infants' lives.
And that climate just produced chaos because the medical and legal systems had no uniformity.
The system was completely fragmented. You had these debates raging about inconsistent brain death criteria. a total lack of defined legal frameworks for non-viable infants, and intense pressure from the media.
And that created public mistrust.
A huge amount.
Yeah.
And it significantly stalled progress in neonatal transplantation. The source material argues that these specific limitations, the physiologic instability and the chaotic donor ethics, are both resolved for IUHT today.
So how has the modern ethical and legal environment fixed those historical problems? What makes IUHT more tenable today?
The change is institutional. Today we have standardized, legally uniform criteria for brain death across the entire United States, detailed by authors like Truog.
And crucially, we now have the mandatory involvement of organ procurement organizations, or OPOs.
Right. The OPOs act as independent third parties. They ensure there are clear, regulated donor authorization pathways, they independently verify death, and most importantly, they prevent any conflicts of interest between the clinical team caring for the recipient and the team involved in donor procurement.
The two teams are walled off from each other.
Completely.
And what about the fetus as a patient? The IUHT framework has to acknowledge fetal candidacy for any of this to be legal.
And it does. The current ethical framework set by the Organ Procurement and Transplantation Network and the United Network for Organ Sharing, that's OPTN-UNOS, they explicitly permit listing a fetus for organ transplantation.
So the fetus is recognized as a patient.
The OPTN Ethics Committee has acknowledged the fetus as a patient with its own distinct clinical interests. This is a crucial regulatory precedent. The institutional barriers that Leggins and Powers were writing about decades ago, they're just not in place anymore.
Okay, let's talk donor sources. IUHT requires a reliable source of viable hearts. What are the pathways available today that meet these stringent ethical standards?
IUHT can draw from multiple regulated pathways. The first, and this goes back to the baby Teresa case, involves anencephalic infants. Following those historical debates, standardized protocols have been developed, as detailed by Paris and others, recognizing them as potentially eligible donors once established death determination protocols are met.
What other sources exist?
We also use structured protocols for donors after circulatory death, or DCD, in perinatal settings. These protocols are incredibly tightly controlled to ensure that all efforts to sustain life are ceased before organ recovery is even initiated.
And a third category.
Finally, there are organs from conditions that are incompatible with life, where some other anomaly renders the fetus non-viable, maybe severe pulmonary hypoplasia, while the heart itself remains perfectly viable.
And there's a unique ethical advantage to doing this intervention in utero, isn't there?
The paper makes a really powerful argument on this point. It says that IUHT removes the element of neonatal suffering and emergency desperation from the equation.
It's not a frantic last-minute procedure.
Exactly. By intervening with placental support while still in utero, the entire procedure can be elective, coordinated, and controlled. And that stability, coupled with modern oversight, provides a far stronger ethical justification than the frantic, unstable setting of historical neonatal heart retrieval and implantation.
So the ethical and technical plausibility have to be proven through a phased approach. Petrikovsky and Nemotin outline this rigorously staged translational plan.
Right, and it has to adhere strictly to contemporary fetal surgery standards. This roadmap is where the conceptual engineering meets preclinical reality.
And it begins not in the OR, but in the lab.
In the laboratory and the engineering bay. Phase 0, engineering and ex vivo validation. This phase is all about addressing the major conceptual tech hurdles before any animal work can even begin.
Let's revisit the technical milestones here, focusing on that miniature perfusion circuit, the PSMU.
The PSMU, the placenta synchronized microperfusion unit, has to be validated. We are talking about engineering a pump that can deliver flow rates as low as 20 milliliter per kilogram per minute with zero pulsatility and minimal shear stress.
All while maintaining that strict priming volume of 40 milliliters or less.
Exactly. And the validation requires demonstrating perfect hemocompatibility, that the materials in the tubing and oxygenator don't cause hemolysis or coagulation, and successfully mimicking those low fetal oxygenation levels to prevent the hyperoxygenation injury we discussed.
It sounds as much like a micromaterial science problem as a mechanical one.
It absolutely is. The surface area of the tubing has to be minimized to reduce inflammatory response, and the flow dynamics have to handle the high viscosity of fetal blood without damaging the cells.
And beyond the PSMU, phase 0 is about that rapid anastomosis workflow.
Right. Teams have to rehearse using end-to-end couplers and microsutures on vessel models, proving they can reliably get arterial and venous reconnection done within that 8 to 10 minute warm ischemia goal, under pressure.
They also mentioned rehearsing hybrid access in high-fidelity mannequins.
Yes, and this is crucial for optimizing ergonomics. I mean, just imagine coordinating a robotic microsuture procedure while you're also maintaining hysterotomy stability, uterine cooling, and fetal monitoring all inside this narrow fetoscopic field.
It's incredibly complex choreography.
It is. Rehearsals in perfused uterine models are essential to optimize that multidisciplinary choreography for a successful, swift operation.
So once the engineering is validated, we move to phase I and IB, preclinical validation. Why is the fetal lamb the first necessary model here?
The fetal sheep model is the unequivocal gold standard in fetal cardiac research. Its size is typically comparable to a late mid-gestation human fetus, and its fetal circulation physiology, including the ductus arteriosus and foramen otole dynamics, is nearly identical.
It's the ideal physiological testbed.
It is. And the success criteria here are rigid. You have to achieve that less than 10-minute warm ischemia time, and you have to confirm functional, stable integration of the graft for a minimum of 24 to 72 hours survival while still in utero.
And then the jump to non-human primate validation in phase IB. Why is that step necessary?
Because while the lamb is physiologically similar, the primate anatomy and, more critically, the cross-clade immunologic behavior are much closer to human models. We need to test how the maternal immune system reacts to the anesthesia and the procedure itself in a near-human model.
So you're verifying maternal tolerance in the surgical workflow before you'd ever consider a human trial.
Exactly. And the authors mandate strict success benchmarks here, 50 to 70% procedural success rates, and confirmed maternal safety that's equivalent to existing open fetal surgery standards.
Okay, next is phase 2, tolerance first trials. This is the Human Immunologic Foundation, a critical risk mitigation step before the actual heart transplant.
It uses the fundamental principle of IUHT, the Tolerance First framework, which was championed by researchers like Flake and Zanjani, dictates that you test for immunologic success before you take the major surgical risk.
So what does that involve?
Phase 2 involves the injection of donor hematopoietic stem cells, the cells that produce immune cells, into the human fetus, probably between 18 and 24 weeks gestation.
And what are they monitoring for in this phase 2 trial?
The main objective is to monitor for stable microchimerism. If the fetus successfully integrates those donor cells and demonstrates that they're persisting over time, it provides compelling evidence that you've successfully accessed that tolerance window.
So only after you've confirmed immunologic acceptance would you move forward?
Only then would the clinical team even consider advancing that specific recipient to the full organ replacement in phase 3. This strategy avoids the surgical catastrophe of organ rejection if tolerance isn't achieved first.
We've mentioned the gestational window a few times. Why is that age, 28 to 32 weeks, so critical for the actual phase 3 operation?
It's this narrow window defined by a necessary balance. It's a balance between surgical size and immunological opportunity.
Okay.
Below 28 weeks, the fetal heart may just be too small and structurally fragile to tolerate the complex manipulation and precise anastomosis required.
And above 32 weeks?
Above 32 weeks, the fetal immune system is rapidly approaching maturation. The risk increases that the tolerance window may be closing, which makes achieving that stable microchimerism less likely. So 28 to 32 weeks is the sweet spot. It maximizes both surgical feasibility and the immunological advantage.
And finally, phase 3, the first in human protocol and governance. The inclusion criteria for this have to be incredibly restrictive.
They must be. The procedure is only ethical if it's offering hope where none currently exists. So inclusion criteria are strictly limited to lethal congenital cardiac malformations that are fundamentally incompatible with postnatal life.
Conditions where the infant is certain to die shortly after birth, no matter what.
No matter what advanced neonatal care is provided, yes.
Describe the final proposed surgical protocol for this phase 3 trial.
The protocol mandates a hybrid approach. Fetoscopic guidance combined with a minimal microhysterotomy, strictly less than 1.5 centimeters, to get access to the fetal thorax.
And the key steps.
The key procedural steps include fetal cardiac bypass using the validated PSMU, opening the fetal sternum, excising the native heart, and then performing those rapid anastomoses of the donor heart, ensuring that warm ischemia time stays under 10 minutes. It's a high-stakes, tightly scripted procedure that demands a level of multidisciplinary team readiness that very few centers currently possess.
Given the immense complexity and the maternal risk, what kind of governance structure is required to make sure this is done safely and ethically?
The structure has to be beyond reproach. You need IRB approval that explicitly emphasizes maternal safety primacy. a mandatory independent data and safety monitoring board, or DSMB, is essential.
And that board needs to have predefined stopping rules.
Clear, early stopping rules for any instance of significant maternal compromise, uncontrolled hemorrhage, or uterine instability. Furthermore, mandatory reporting to the NAFTA registry ensures full transparency, so the procedure safety and outcomes can be directly compared to existing fetal surgery benchmarks.
It's a robust governance system.
It's the direct institutional answer to the chaos of the 1980s.
Before we move to our final takeaways, Petrikowski and Nemotin included this really fascinating and cautionary note on literature integrity. They specifically highlight the danger of AI-generated misattribution in a fast-moving field like IUHT.
This is a vital point for any researcher operating at the cutting edge. They cite a specific example, a non-verifiable citation that's attributed to a Chamberlain et al. Concerning fetal cardiac transplantation.
And this reference has appeared in AI-generated bibliographies.
And secondary sources, but it doesn't correspond to any verifiable peer-reviewed publication in any official medical archive. It just doesn't exist.
So a large language model essentially created a plausible but completely fictional reference, and that fabricated information is now circulating as if it were fact.
Exactly. AI models, when you task them with summarizing or generating literature reviews, they can often hallucinate citations by synthesizing common author names and established topics.
And in a sparse emerging field like IUHT,
where the difference between established science and a translational concept is very narrow, chasing these phantom references, wasting time and resources trying to verify claims that don't exist, can actively undermine the verifiable translational pathway.
So the takeaway is that for high-stakes, life or death research like this, rigorous manual verification of every single source, cross-checking against primary archives, is absolutely non-negotiable for scientific integrity.
You can't just trust the algorithm.
Okay, so this deep dive into the work of Petrikowski and Nemotin has really shown that in utero heart transplantation is not science fiction. It's a meticulously planned framework built on decades of surgical and immunological advancement. Let's review the key conclusions we can draw from these sources.
I think IUHT's scientific plausibility really rests on two biological pillars. First, the gestational environment's circulatory cushion, the placenta, which gives continuous support and eliminates that high-risk mechanical bypass.
Right.
And second, that unique immunological window of opportunity in the mid-gestation fetus, which offers the potential for true long-term tolerance and the avoidance of that debilitating lifelong immunosuppression.
And furthermore, the ethical and regulatory barriers that haunted those earlier neonatal transplant efforts,
Right.
the ambiguity of death criteria, the lack of third-party oversight, those have been institutionally resolved.
They have. Today's OPO and APT-UNOS frameworks recognize the fetus as a legitimate transplant candidate. That positions IUHT squarely within the boundaries of modern medical governance.
So IUHT is the logical and necessary next step. It integrates the original anatomical proof of concept from Demikoff with the technical precision of modern fetal surgery that's been documented by people like Adzik.
The technology is conceptually developed. It simply requires that rigorous engineering validation that they outlined in the Phase 0 roadmap.
So we've established that the success of this whole translational framework hinges on the invention and validation of these conceptual engineering devices. The placenta-synchronized microperfusion unit, the fetoscopic microsaws, the rapid anastomosis couplers. These innovations are what's required to anchor IUHT as a clinical surgical discipline. So here's the final thought for you to consider. If whole organ replacement becomes a routine possibility in utero, what is the next great conceptual leap required in fetal medicine?
That's a great question. I think the immediate next challenge would probably shift from replacement to regeneration. If we can replace a failed heart, what about developing techniques for in utero regeneration of complex tissues? Or maybe even genetic editing to prevent these lethal conditions from developing in the first place?
So the goal shifts from fixing the failure to precluding it entirely.
Exactly.
A fascinating thought. We invite you to consider that broader scope of prenatal repair that might follow once the most critical challenge, whole organ transplantation, has been overcome. Thank you for joining us for this incredibly technical and expansive deep dive into the future of fetal medicine. We'll see you next time.